Okay, so today we have an experimental presentation from a smartphone. That's one of the differences. But go ahead, Jordan. Please.
Yeah, of course. Thanks. Thank you. Thank you so much. My name is Jordan Clemsen, and today I'll be reviewing briefly the paper Genotype and Phenotype Differences in CADASIL from the Asian perspective. I hope you enjoy it as much as I did. That's great. Well, you got it.
I'm just gonna.
Yeah, thanks. Oh, that's a good idea.
I don't think there's anything I can do to fix the.
Yeah, no worries. I'll shoot it right there. Essentially, so what CADASIL is, it's a well short for cerebral autosomal dominant atherosclerosis with subcortical infarcts and encephaly, which is basically just a really fancy way of saying that it basically is progressive mini strokes that occur when the brain essentially just kind of has a little junk in the blood vessels that then break down and then which the debris and the necrosis continue to clog up other blood vessels in a continuous feedback loop that deterioration and beautiful sweet entropy. So as we see that, we also know that it's a constitutive dementia, which should be underneath vascular dementia itself and Alzheimer's. So there's a relation. What's interesting about this connection and why I'm bringing it up is because it's supposedly a genetic link, and one that we are more certain about the genetic correlation with, and which should distinguish from other constituents of dementia itself. Some of these comorbidities are vast, as you can tell from the MRI slide. There can be different areas of the brain where these lacunes or white matter hyperintensities and even microbleeds occur, which then can affect a vast number of things. Symptoms can range from anything to mania to memory loss, cognitive decline, hallucinations can occur. There has been quite a bit. Typically, though, which in general, there's mostly psychiatric depression, memory loss itself, motor impairment, and typical with dementia. Although that can vary severely depending on where it's at.
Come on in.
And symptoms as can occur are similar to that as well. Atherosclerosis and diabetes are actually immensely associated with CADASIL itself, just because it just the only thing that changes is what actually is clogging up the blood vessels in the brain. Whether it's plaque, I've seen stuff that personally I have noticed a wild type HTT that has looked very similar to clogging up the plaque itself too. So it just the comorbidities usually change what is clogging up the plaque, and or if the lesions in the brain are actually causing the disease as a symptom. There's multiple tests to actually go over this disease. Just a brief overview of CADASIL itself. There is usually people use a skin biopsy, which has been genetically linked to determine the gene of association, or the main gene of association, I should say, which is Notch3 themselves. There's also MRI diagnosis as well. So there's a genetic component, an imaging component, and there's also a clinical diagnostic for dementia, but that's usually not there. It's across multiple fields itself. The Notch gene itself deals with epithelial growth factors throughout the body. The Notch3 has been known to associate growth factors in the brain with the blood vessels themselves, which is why typically this is associated with this disease itself. Let's get the next slide.
I've got a question. So
Perfect.
you said there's an accumulation of plaque that can be symptomatic of it,
yes.
But you also said is it actual plaque accumulation, or is it the thickening of the arterial wall? Right?
It can be that as well.
It's both. Okay.
Yes, it can be that as well. It's if you one of the overarching things I'm gonna discuss throughout this review of this paper, and I think what this paper is trying to allude to, once I get to the paper, besides the background information, is that this is a very poorly understood disease itself, but it has the potential to be very interesting. Answering quite a bits of questions in dementia itself. And more specifically, even so to the genetic components and to a better understanding of, spoiler, white matter hyperintensities. So that essentially is going to be the outcome. So there is benefits to each field, but it is very poorly understood at this point, besides just the vagueness of a deterioration of the brain through necrosis itself and asphyxiation, hypoxia of blood vessels. Which is pretty vague itself, but and as you can imagine, manifest in many different ways. Is there any other questions about CADASIL? If not, I'll ask a question. None? Okay.
What was the acronym again?
No, what? Pardon?
What was the acronym again? Can you say it?
Oh, yeah,
I
cerebral autosomal, so it's a sometimes I can't. Cerebral autosomal, cerebral autosomal dominant and then the cerebral autosomal.
You don't need to.
Yeah, sorry. Trying to remember with subcortical infarcts and leukoencephaly.
Yeah, it's a hard one to remember.
Yeah, I know. It's like I always get mixed up on the D. It's dominant, I think.
It's
dominant.
Autosomal dominant. Yeah, thank you. Thank you. That one always, yeah, I wish they would just swear it's called CADASIL. Subcortical infarcts. Okay, so true or false, can CADASIL cause manic episodes? Anybody to shout it out?
True.
True, sweet, that's great, that's correct. So, good, yes, pay attention to the can, because it's part of many symptoms. There's been a case study I think I recently looked at and want to cite my meta-analysis that really kind of goes over the mania and why they think it's causing mania itself. Okay, so now that we've discussed some of the background information of CADASIL itself and an overarching view, the gene itself, which is discussed a lot in this paper, is Notch3. Notch3 is essentially the gene associated with the subcortical infects and the pathogenesis of the disease, the pathology of this disease itself. So essentially, this gene is located on the 19th chromosome. It's on point. You can see it's located right here itself on the P13.2. Typically, it interacts different axons itself. The axon ranges significantly, there's quite a bit of variability. Supposedly exon anywhere between exon 2 and 23 has been associated with the mutations or disease. However, there have been instances recently of exons being as high as 25 and 33, which is interesting. So it kind of defies the rules that we've set up and raises questions. How this gene typically works is this part usually is mostly for extracellular interactions itself. So it, once it gets kind of into where it wants to go, then this part is the part we really care about. I guess this part kind of matters too because it has to get where it's going. But once we get there, the epidermal growth factor itself and the repeats that are throughout here, break apart, and then they actually interact within the transcriptome itself to actually influence other gene expression. So this is like a multi-step interaction that occurs. So like a slight deviation here could really kind of throw a wrench in a whole sequence of events that occur that have for considered healthy protein. The mutations here this is usually where we see the mutations occur because again, if it happens here, it's probably not going to get where it's going. And it's not really going to matter much, right? So we see usually mutations here in the in EGF and the R. Usually what we see is cytosine added or lost. But obviously that can vary itself. So
Is there research about which kind of mutation does result in the manifestation? Is there research about that?
I would yes across multiple. So I'm going to review each parts of this paper and really kind of imply something in a direction. But each paper itself actually talks about a different type of mutation that they've had. I think that's actually one of my points here. Right here. So this cohort was essentially these people. This study and the reference, which is really interesting, was the people in. This is just in. This is the Germany one, I think. My slide. Yeah, this is the German one. So this is Germany itself. So essentially, what this paper did is they went through and they they basically did skin biopsies on 125 patients, German patients, in hopes to review, use qPCR and other Western blot techniques to review the spectrum of what the gene itself actually produced and just report that. And so what they did is, well, actually, I should ask for what they did. The main test they did to extract this was from the epithelial cells itself. It's a skin biopsy test that could be with CADASIL, and that usually tells you the gene association. So that means if you're genetically positive or negative, usually is what it comes back as. Is everybody here familiar with the center dogma at all? I'm assuming, right? Okay, so I'm going to assume that until otherwise. So essentially, there is clearly a different a variation between the translation and the transcription that occurs within the production of this gene and part of the protein itself. And what they reported was 54 distinct mutations. So there isn't just one different mutation as we were thinking. They were reporting that they found in these German patients that there's 54 variabilities in the way that the notch gene itself actually changed. And that's pretty huge. 96% of these were missense mutations. Missense mutations are in the translation phase where the gene actually produces a different protein itself. So it's a slight variation in the protein because of a different amino acid that's added. So that obviously can kind of throw things off in a macro sense of the disease itself. They did report some frameshift mutations, although they're very few. They reported other ones too, but usually what the mutations occurred was different types of missense mutations. So like it was a different polypeptide that was expressed. Here's the whole list. I know it's kind of hard to see. I was questioning whether people would see this. I know I couldn't, but I mean, I'm blind. You can't. Okay.
No, it's way too small.
Good to know. I was like, I can't. Maybe they can't, especially if it's on a big screen. So essentially, what we're looking at here is they listed all majority of these different mutations. What was inserted into the gene, whether it's thymine, guanine, even adenine, or anything like that, and any other complex nucleotides. They looked at the frequency of how many mutations there were. I'm just going to shout it out and someone else can see it because I get it. The range is 20, and the lowest is 1, but there's a whole bunch of ones, so there's tons of variation. And they also reported where the repeat was and what was replaced, what the nucleotide was replacing. And what they found was that the proteins that were actually produced itself were the highest amount of protein too is 32.5% of this EGF on protein 4. So those are the highest ones there. Excellent. And so I'm going to start off this journey of this variation in genetic diversity up in Finland. So there was this Finnish study that then decided that wanted to report their patient population of CADASIL. And so they took 294 patients with a diagnosis of CADASIL. That was actually important. They were diagnosed from clinical what was it? From the clinical record. And tested them again from anywhere from the range of 1996 and 2019. A lot has changed they reported that there was multiple changes that occurred in the disease and the definition of the disease itself. So this did alter some of the data. However, they discarded the ones that were not relevant to the changes. What they found was that the Argentine itself, protein was the most, that haplotype mutation and protein expressed was the most prominent with 68%. There was different alleles they found throughout Finland, as noted from the 68%, and this manifested itself in different clinical symptoms, whether anywhere from memory loss to even hyperinsomnia, which was really interesting too. So it varied, and they reported the patients reported different symptoms from the proteins themselves. The table here, I'm assuming you guys can't see this one either, is essentially the p-values associated with mutations in the proteins that they had, and with the age onset risk, epilepsy, anything from psychiatric symptoms, anything else, and yes, it's okay. and essentially the p-values associated with it. The p-values aren't very significant if you can tell in certain areas, but I want to point out that those are usually, there's probably other mechanisms and co-confounders influencing that. So essentially what my takeaway from this study was that Finland is very varied, although they have the most dominant form of the protein expressed is PRH133 and what's cytosine. The next one I'm going to discuss is now we're moving closer to China. If you can think about it geographically, that would be helpful too. So there's 21 patients in this Chinese study that used PCR and effectively did the similar things that Finland did, but in a Chinese population itself. They found 9 different heterozygous point mutations, which is really interesting. They even cross-referenced it to previous haplotypes that were with other studies, including the Finland one. And they noticed different changes too, but they also noticed a different prominent they had a different haplotype that was more popular than Finland itself, which is the main takeaway I want to you take from this slide, as well as the different types of symptoms that it manifested, as well as headaches, seizures, cognitive impairment, and ischemic strokes, which is the kind of strokes that we see, the progressive mini strokes that I referred to earlier. So I have to make that smaller. There we go. All right, I'm gonna have to move this down. Okay, and the next study I'm going to refer to is a Korean study of spectrum of NOTCH mutations in Korean patients with clinically suspicious cerebral autosomal dominant seriopathy with subcortical infarction and levoencephalopathy, which essentially, and in this study is 156 patients with clinically suspicious and not previously diagnosed with CADASIL were tested with qPCR, Western blotting, and other I forget what other one they did, but they did another genetic testing as well too, and they reported their findings, noticing proposing that R544C, that variant was the most prevalent. They also noticed that I want to point out, and let's see if I can open it down. I think Zoom is waiting. Okay, well I zoomed in on my phone. So if you can't see here, what they did report is that some of these variations and the most prevalent variation itself actually differed based on the islands themselves in Korea. So in the Jeju Islands, there's actually a different amount. There's a different most commonly associated variant, and as opposed to other areas in Korea. And then they also, just for even more replicability and to get the point across, they reference other the other most popular points themselves across other studies, whether it be France, UK, Netherlands. For the sake of my presentation, I tried to narrow it down and make it more streamlined. If you can see here, the numbers are the numbers of exons are where it's located and where the gene is located is different on for the Korean patients as they are for patients of other nationalities. And this leads, and then finally, the final study I'm going to do, referring to national within the nations themselves, is going to be the Genotype and Phenotype spectrum of CADASIL in Japan, which is essentially the Japanese study where they did a very similar structured type of study where they tried to monitor the patients themselves. Of 215 patients, they used a similar skin biopsy test, and then they reported their findings in the most prominent variant of these alleles with the location of where the gene actually is and changed in a mutation. They proposed that they reported that there's different alleles by region. They actually reported too that there is different features in MRI, which I will discuss in a little bit more detail later. And there's also, which I thought was really interesting too, is that there's different features also immunostaining that they did. So there's actually quite a lot to unpack here. But they also noticed that from their population of 215, the mutation of RG133 with cytosine had much more white matter had greater densities. The graph I want you to see here is the most prevalent allele based on the region of Japan. And you can, and I just want to point out that as you can see, the more isolated regions kind of have more more variation and more deviation from the rest of the group in the mainland. Okay, any questions about my genetic session at all? Anything about that? From there? Okay. What does EGFR stand for? Whoever shout it out. I know. It's hard to say. Epidemial growth factor repeats. So that's the part of the tail in the notch that I pointed to. And that's usually what's changed because once it enters where it wants to go, it kind of breaks apart and attaches to different sites in the DNA and RNA. It's DNA. Don't quote me on that. And then essentially influences the expression that they can produce more proteins. Okay, and so this kind of builds to where I'm going to, where there is a huge variation in the gene itself by region. I didn't go over Italy, but I mean, I think it's kind of superfluous at this point. Exons 2 and 6 are the most common in Europe, associated with 68 and 60 to 80% of the mutations found. So CADASIL in Europe most likely will have a mutation of a notch-3 gene on 2 to 6. Exon 4, this differs a little, it's slightly different in the Germany, United Kingdom, and France, which if we think about it geographically, is kind of this area. But if you go to the more Mediterranean area, you do get that's supposed to have the variant attached to it too. I don't think that it transferred when I changed the design. That stinks. It produces a different allele, but still on axon 4. And this different axon itself actually varies slightly, but still on the same axon itself. Once you go to, yeah, that did not transfer. Axon 11, 48 and 80% are changed in Korea. It's in China itself. That's supposed to have China on the end of it. And so as we go further along a geographical effect, we see it changes axon completely, is my point there. Exon 11 mutation is even rare in Japan. So once we get all the way over to Japan itself, the same axon that was present over here is now really rare itself, which is a point I want to make. And 70 CADASIL participants in Japan, there's 0 axon 11 mutations. So as previously cited in this in the overarching meta-analysis review itself. And so that kind of we just go to this short area and it changes that much. Also, I want to point out that the Japan variant, and the paper does say this too, produces a greater disease severity itself. So it actually produces a later diagnosis of the disease, but much more severe. It's much more impactful in a clinical setting. So you actually end up with greater cognitive decline along the line, more serious, quote unquote, serious symptoms like seizure and stuff. So the severity does actually increase with the Japan variant, which is interesting too. And where am I going with this? Well, it seems like if we move back to biology too, this seems to really mimic the concept of a founder's effect. And so what is a founder's effect? It's basically when the genetic variability decreases, heterogeneous disorder increases because there's a lot of recessive genes that all of a sudden have more recessive genes that can pair up with, and we see more expressed a disease itself because we're similar, and because we're similar, the similarities pair up, and then we have an increased chance in offspring. Whoops, sorry. This has been previously experienced. Some of the concepts where this applies is the California condor. Back in the 1980s, they and the paper does describe this a little bit too. The California condor in their citations. The California condor was essentially just really densely grouped together, and it was only 2,000, I think, reported back according to the Wildlife Conservation. So they were taken to the San Diego Boston Animal Park, and hopefully bred together, but there was a lot of genetic diseases present, multiple, because due to the limited population. Limited population, limited environment, and limited genetic variability, which increased the genetic drift significantly to the point where there was a lot of just chaos. So this was monitored, and then currently, since the 1980s, we have been able to help with the species, but that's a famous example. Currently, and then a more recent example is the California cougar, as there is a highway, I can't remember the highway itself, so if anyone remembers the highway, please shout it out. But there is a highway out in California that is basically cut and stopped all the cougars themselves from actually migrating, so they've been stuck in, I don't know if anybody's been to California, but the traffic's annoying, and I don't think a cougar would want to walk through the highway. And so they have interbred to the point where their genetic variability is decreased, and their disorders are pretty bad, and some of them have actually been infertile because of it. There's been a lot of talk about redoing the highways just to stop the species from going extinct from this very similar concept itself. My point is, why I'm talking about this is there seems to be a very similar founder effect occurring, especially which would also describe, which the paper describes in much more detail, but also other studies that I cited have suggested too, which would describe the isolated islands in Asia, why the disease is so much more significant. And it's assumed that this is a founder's effect. I want to point out a couple things about this concept, is that there is it's an increase, it's a type of genetic drift that does not have to do with natural selection. So the fittest are not selected. What actually is selected is a number of things. It's different from a bottlenecking effect. So in a bottlenecking effect, essentially think of a bottle itself, and something comes in and selects on these things that is not natural selection. That's the important part I want you to take away. For founder's effect, some of this population is picked up and put into another plate. And because this whatever it is picks it up and puts it into another area, the ratio changes, which then alters the genetic frequency and could decrease genetic variability due to the autosomal and recessive natures of the genes of the alleles themselves. So that's what I want you to take away from that. It's very it differs significantly because this population goes on to live, even though this one just got sprinkled somewhere else and can't really interact with this previous one. Okay, any questions about the population ecology section that I have? Anything like that? Any questions? Okay, if not, I have a conceptual question to ask. So let's say Jordan catches all these metagross that are in Pokemon that are shiny, so they have a different allele. Is this an example of the founder effect? Why or why not? Anybody want to shout it out?
Depends what you do with them, I suppose.
Yes. If I'm going to, well, I'm gonna say I'm gonna keep them and just have them interact. How about that? And they're not gonna interact with the wild guys because they're I declawed them. How about that? So now they're with me. So I would say yes, it is. If it would be a bottlenecking effect, if I just battle the rest of the metagross and then just harvest the XP, then that would not be a that would be a bottlenecking effect itself. They're both forms of genetic drift, but they are not natural selection because this is not a convenient event. This is an environmental effect, like a thunderstorm or something that is coming in and altering the population itself. So about that part, now I want to get to the part that we're probably more interested in, clinical and genetic aspects of CADASIL. And so we see that there's typically in healthy notch variant, there's 34 epidemial growth factors in the gene itself. So that's like typical. Obviously, these missense mutations are the most commonly reported. So that's what we've probably been referring to as a notch. Usually what this does and how this manifests it is it actually increases in excess in notch protein itself. So whenever these things are breaking off to and attaching, what it usually does is create more notch, which is somehow influencing the vessel breakdown in the brain. So I tried taking a picture of the pathway here and the report that followed, but I think I'm going to skip this for the sake of time. The nonsense mutation, there have been nonsense mutations reported. They typically get cut off, and the manifestation actually differs. The first frame shift mutations were actually reported in 2007, so those are the least likely. We've reported, as previously mentioned, the type of mutation itself is more common in Finland, which is interesting. So usually and I discussed what happens in the pathway, it's the molecular pathway itself. Usually what happens is this third cleavage on the disulfide bond over here that is cut. And then this part essentially is this part, this is cut right here. This part, and this is cut because this is the excess DNA that is then just there that check the rest of the DNA, like an intron itself. And so this part is the part that's expressed more into the and then breaks apart and then attaches itself to other genes and then produces the protein itself. I think I discussed that a little bit earlier, but just good to discuss that. So then finally, how does this affect the imaging and in China itself and the clinical symptoms itself? So this Chinese study that the paper then cites, I think describes it pretty well. So this study basically had 112 patients. The mutations were from exon 2 to 24, and then this mutation, and then what they did is they use qPCR, Western blotting, and also imaging techniques too to register clinical symptoms as well. And what they found is it's associated with a lower frequency of anterior temporal involvement in white matter hyperintensities and a later age of onset and higher frequency of the cognitive dysfunction itself. So from the Chinese variant, it looks like there is a later age, a higher frequency, and more symptoms typically. And then there also the location differs. That's one thing we'll discuss later. Serum microbleeds were detected. 87.5% of patients of this 112. But then they were most frequently observed in the thalami itself, followed by inferior, eyes not the best, inferior regions. I'm gonna let you do the rest. If you can see it, I'm assuming you can see that, right? Okay. And here, and they report their findings here. You can see that I think I was supposed to point. Here's where that is the oops. Right. This is where the microbleeds were most centered, and then I think it was B, which is where the lesions were most likely centered. So in conclusion, the imaging difference, in conclusion, the whole entire review article, it discusses that Asian populations with CADASIL have a lower anterior temporal pole positioning. With EC if they use a different type of imaging, there's more white matter hyperintensities. 87 and 93% of this is affected in European population, while if in comparison to Korea, which is directly contrasting that, there's only 45%. What this suggests is that there's a suggested area of mutation that is then correlated to an area of where the white matter hyperintensities manifest. And why this is important is because if you see white matter hyperintensities themselves, it's kind of where you avoided a minefield, if that makes sense. Because really what happens with the disease is when you get the bleeds themselves, that's the area that affects the symptoms itself and severity. So we had a patient 2 weeks ago who had white matter hyperintensities, but was doing amazingly on his cognitive, his MoCA and everything. And I remember thinking about it and thinking this person's probably fine, and looked at his scan and was like, whoa. And the reason is because these white matter hyperintensities were not in the proper region, or in a region that would have significantly debilitated him. So this is clearly important. It suggests that this mutation was probably in a safer area that is probably going to hurt him more later, but will help him. But for now, it's going to make his cognitive assessment relatively normal. Any questions about what I just discussed in the MRI section? Anybody? Show them out.
Is there any indication of a genetic marker for the spatial placement of these white matter hyperintensities? I haven't seen anything like that, but
So it's suggested. So it's just because of but there, it's not been isolated enough for them to confidently say that there was, but it does seem like there's an association. Good paper, future paper probably. I think it'll be great because then you could actually like, I think from a clinical standpoint, the skin biopsies could actually be much more directly communicated to the patients. Neurologists would probably say, oh yeah, you're probably gonna do this and you're probably gonna hurt later. But for right now, you're gonna be fine. So I would, if you have like any vacations or anything fun you want to do now, get it now. Versus like if they're debilitating now, you're gonna probably have to quit your job, but you're gonna, it's gonna be progressive. It won't be that much different to expect. So that obviously helps help from a healthcare setting much better. One question I had is what CADASIL MRI biomarkers are different between Germany to Japan? Yeah, so that right. So there is a lower frequency of white matter hypertensities in the Asian population. Also, the disease severity is increased much more in Japan, which could imply due to the isolated events of the islands themselves actually creating a adverse effect and accumulation of recessive genes that probably produced are suggested that it produced a more severe disease. And that is my presentation. I don't see the next button, but I'm gonna stop sharing. Thank you so much. Is there any other questions? That's all I had. Any other questions? Oh.
I think people are maybe thinking, because this is mostly an ML group, right? And some people are not as familiar with some of the background.
Yeah, that's why I tried to like do a slide in background stuff.
But
I can do the next paper. I promise will be more interesting with that. I can do that.
yeah, are there a lot of like multimodal analysis methods incorporating genetic and imaging?
So what I've seen is that so doing this meta-analysis has been frustrating actually for that reason, because I see that there's a lot of conflicting. They're either much more on the MRI side, or the MRI is mentioned, but then like for example, for meta-analysis, this has been really frustrating. They don't describe how they defined like matter hyperintensities, which is exactly right. Which is a little frustrating. They just say white matter hyperdensities. There's one paper I threw out yesterday because it used interchangeable words from anything from abnormal white matter to white matter degradation to white matter hyperintensity. And that and using that interchangeably and then reporting their variable, I have no idea what this means. So what I've seen is a miscommunication between there. There's been a couple, but they've been much more MRI focused. They haven't been, they don't really incorporate much qPCR or Western blotting. But that's a really good question, and one that's been really frustrating because it's like, how do we concatenate these biomarkers if I don't even know what this person means? Sometimes they don't even report their software. So
and what have you found in terms of functional imaging derivatives, right? Because white matter hyperintensities are a structural, artifact, right?
Yeah. I will tell you as soon as I meet with the statistician next week. That's a good question because we're just.
So you haven't found any other works looking at any functional derivatives?
Nothing functional, no.
Surprising.
Yeah, I kind of was a little bit too. There was it was mostly just the location of the microbleeds because they're kind of more worried about the microbleeds right now because that's where they see the disease. I think that's where people are focusing. That's my understanding. But I think the white matter hyperintensities are poorly understood currently. But I think they should be something that we should probably. I think there's something that influences the disease in a way that we would really help our understanding if we research it. That's what I think would happen.
More methods question. Someone else has a question?
Yeah, sorry. Anybody else have a question? Sorry.
Well, another more methods question then is: do you know if all of these studies are manually labeling their lesions, or are they using automatic labeling? Because that's something we looked at was using MeshNet or a 3D CNN or the UNet style segmentation models.
So most of them I've seen have actually been using So the ones I have seen, they've used software packaging. Back I look at my meta-analysis really quick.
Yeah,
I did write
is it just
that.
Free Surfer, because Free Surfer has
A
legal
lot of, yeah,
segmentation.
there was a lot of free surfer. There was some SPM.
Mhm.
There was definitely, I think they reported SAS, but I think that was for the statistics part.
Yeah, I don't know.
Yeah, I'm pretty sure, because I don't think that would die. I don't
Your
think so.
segmentation in SAS sounds.
It's hard to like say, what is it? It's hard to look for software itself and then not associate what the software is, because it's a lot of free surfer, different versions of free surfer though, that's probably something that's pretty significant. I did notice that there's different versions, but I doubt that would change very much, because the algorithms themselves don't really change as much, unless you in greater amount of time. Oh, and then one thing that's changed though, not in these papers as much, because these ones are more recent. But in some of the ones that have been in the meta-analysis, the Teslas have changed significantly. I think I have one back in 1999. That's one Tesla that I really just kind of think I should throw out at this point. And then there's 1.5, there's 3, and there's 7 more recent. So that's been something that I think really needs to be addressed. But yeah.
Well, it would certainly, are you doing like an actual meta-analysis or just like a review?
Well, it was intended to be an actual meta-analysis. However, the field is very vague itself, and
Well, is the data available is the issue.
yes, there's a lot of these papers that I feel like I have to reach out to the authors themselves and ask very specific questions for clarity. That's been my point. But that's the disease, that's the progression, and
Cool.
yeah. Thanks.
I think that's it, folks. Yeah.
Yeah, we're good for today.
All right, we'll stop the recording and let's see, how do I stop this?